everyone, my name is Kimberlyn Petty, and I'm going to be talking about flow cytometry today. Flow cytometry is an application that I use all the time in my current career, and I think it's actually just a really cool application, and I'm really excited to talk about it with you guys today. So what is flow cytometry? Flow cytometry is using fluidics and lasers to analyze and quantify cells or particles. Three components to flow cytometry are first your fluidic system. So this is using most commonly something called sheath fluid, and this essentially moves your sample from either a well or a tube throughout the entire instrument, and then this moves the cells into a single file line, and we can kind of see that in the image here, kind of more, you see the sheath fluid moving in, and then it goes to something called the flow cell. And again, this creates a single file line for the cells so that the lasers can analyze each cell one at a time. And then this is part of the optics system. So this is excitation of light sources like your lasers, uses lenses and filters, and these are what all are used to analyze the cell. Forward scatter measures the size of the cell. So this is a laser that just hits the cell set dead on, straight from the front, and the way it refracts will kind of help the instrument to determine the size of the cell. The side scatter has a little bit more of an angle, and it will hit the cell, and depending on, again, how it refracts, it's able to measure the granularity and the complexity of the cell. Then after it kind of flows through these lasers, the fluorescent signal is collected. Following the optics system is the electronics. So this is what converts the signal that the instrument is reading, and it converts it into a computer or software so that way we can then analyze the data and kind of understand what the data and the lasers and all that stuff is telling us. Quickly, just some applications of flow cytometry. It can be used in clinical and research environments. And what's nice about it is that results can be seen in real time. So this is really beneficial for clinic-based work, as we can see how the immune system is running in real time. So a lot of times, like in clinical settings, they'll run blood work, and then there's small little flow cytometers and kind of like these blood works, so that way they can kind of quickly see how their cells are looking, how the immune system's looking. And then again, some applications of flow cytometry is immunophenotyping, self-sorting, cell cycle analysis, and apoptosis. The applications that I'm most familiar with is immunophenotyping. So essentially, like what you're doing is you're quantifying and you're analyzing subsets and populations of cell types. So you're taking a whole blood sample and you're processing it all the way down to just white blood cells, and then you're trying to figure out how many leukocytes and lymphocytes and B cells, T cells, NK cells, monocytes are in your sample, and essentially you're getting numbers. So we're able to see how medications are interacting with the immune system, how any sort of effects are happening to the immune system. So it's just really cool that it's kind of like a snapshot. So you could take a sample before a treatment or a therapy is done, and you can see how the immune system is. And then you can see again, like an hour later, and you can see how much has changed within that hour. And it's actually quite interesting. And then you can also use flow cytometry to kind of see how long it takes for the drug to kind of work through a system. So you can see how long it takes your body and your immune system to get back to baseline. And so it's just really interesting how that works overall and how we can see it. On cell types, there are things called glycoproteins, and these are called cluster of differentiation markers, or CD markers. So these are unique to each different cell type. If we're looking at the hierarchy on the left-hand side, we can see that CD45 is a marker for leukocytes. So you see your T lymphs, your B lymphs, your thrombocytes, monocytes, granulocytes, those are all examples of leukocytes. And then from there, you can kind of differentiate more using these CD markers. So for example, in this slide, I kind of gave the example of T cells, and specifically NHP CD markers. These are CD markers are unique to different types of species. So what a CD marker is for a certain cell type, what it is on NHP might be different from canine or feline or rat and mouse, etc. So you just want to make sure that you are looking at the right CD markers for the species in which your panel is built for. So this whole slide is an example of looking at T cells. Looking at T cells, we see that the CD3 marker is our T lymphocyte CD marker. And so we're looking at the cell subsets of helper T cells, cytotoxic T cells, and activated T cells. And these all have the CD3 marker. But the different subsets will have additional CD markers that help to identify those. So antibodies in flow cytometry will have a conjugated fluorochrome to those antibodies. And the fluorochrome is what is used by the instrument and by the lasers to detect those antibodies and detect those cells. So looking at the chart here in the middle, we see the cell type and then the antibody that we used. These are all anti-human, I believe, which have cross-reactivity with NHP. So it just doesn't say anti-human or anti-NHP, just for shortened reasons. But your lymphocytes, we see anti-CD45 V500. So this is anti-human CD45 antibody, and it's conjugated to the V500 fluorochrome. So that's your fluorochrome that it's bound to that antibody. And then we can see the T cells, we're using CD45 and then CD3. Helper T cells, we're seeing CD45, CD3, and CD4. CD4 is what's unique to our helper T cells. And when you look at the little illustration on the right-hand side of the slide, we see the half that's T helper cells, and then we see that little red box, and it's CD4, because that's unique to that subset of T cells. Looking at the cytotoxic T cells, we see CD45, CD3, and then CD8, because CD8 is what's unique to the cytotoxic T cells. So basically what this slide is kind of showing you is just that the layering of antibodies in your antibody batch is what it's called, or antibody cocktail, is that you layer them to get all your different cell types and your different cell type subsets as well. Building off what we talked about in the last slide, we're building an antibody panel. So essentially one of the first things you kind of have to do is when you're building a panel for flow cytometry is you need to build an antibody panel or an antibody batch. So you want to make sure your antibody batch has all the CD markers for the cells that you're interested in finding in your sample, all your cells and all your subsets. And one of the hardest things is figuring out the fluorochromes that will work in your panel. So fluorochromes can be conjugated to any antibodies. A lot of different companies will have your CD8 bound to multiple different kind of fluorochromes. They could be bound to FITC is a pretty common one, per-CP Cy 5.5, APC, etc., etc. Like there's just a ton of different fluorochromes that it could be bound to. But the things that you want to look at is you want to make sure that you keep the brightest fluorochromes that can be found on like your more rare or dim populations. So you're going to want to use your fluorochromes that are really bright for like monocytes or NK cells, maybe dendritic cells, or even the subpopulations if you're not really interested in looking at the other subsets. And then another thing you want to kind of think of and keep in mind is your spectral overlap. So you want to make sure that they don't overlap in the channels too much, and you can kind of see that in the front part area where there's a lot of like red and blue. Like you kind of want to stay out of there, and you want to make sure that they kind of separate pretty well. And then that way the cytometer can read it and you can really be able to tell what your populations are. Like CD45, for example, like we said in the last slide, is a marker for all of leukocytes. So it doesn't have to be super bright because you're going to have a lot more cells. And so you want to kind of so you can use kind of a dimmer fluorochrome for that CD marker, and then you can use your brighter ones for, like in this case, like your CD3, your CD8, and your CD4. One of your daily controls that you're going to do is your cytometer setup and tracking, and this is called CS&T. So this is an automated software and reagent system, and so it defines and characterizes the baseline performance of the instrument. It optimizes and standardizes cytometer setup and tracks cytometer performance. This helps to create consistent, reproducible data by offsetting day-to-day instrument variability. Essentially what your CS&T is doing is it's just making sure that your lasers are working appropriately for that day. If you have any errors with your CS&T, it'll tell you that it doesn't pass. The software just won't pass CS&T. And so you'll essentially have to go through and you have to do some troubleshooting to try and get it to pass and see why it's not passing until it does pass. Compensation is another daily control that you have to run. And so essentially you take that whole antibody batch that you made and you're going to create wells per antibody batch. And those are going to be one antibody batch per well in each one of your antibodies. So essentially what this does is it allows the cytometer to read just that fluorochrome in that well. And then you can go through and you can tell the cytometer exactly where you want it to read that fluorochrome. So you're kind of looking over here on this graph to the left, and you see we have per CP Cy 5.5, PE Cy 7. And you can kind of see where the peaks, they kind of overlap each other. So we essentially want to get rid of that. Without getting rid of that, we kind of look at the graph on the left. You see in the first half, it says, or it has the uncompensated. And so you can see per CP Cy 5.5 and then PE Cy 7. So you're looking at the negative, and it's where it should be. It's in your negative gate, your double negative gate here. But it looks like it's both positive for your PE Cy 7 as well as your per CP Cy 5.5. And this is just supposed to be looking at the per CP Cy 5.5. So it shouldn't be showing positive in the PE Cy 7. So by compensating it, you're getting rid of that spectral overlap. And where it kind of is filled in on the left-hand graph, that's where you're telling the cytometer to read it. You want to read it at the brightest point of that fluorochrome. And so once you compensate it out, it looks at like the compensated graph on the right-hand side where you just see the negative and the positive gate for your per CP Cy 5.5. This helps to be able to identify where your populations are. They don't end up spilling over into other gates and other areas of your graphs. And it just makes it a lot easier to identify your populations accurately. Another control that's done is fluorescence -1, and this is called FMOs. So essentially what an FMO is telling you is it's telling you where the absolute negative for your population is, so that way you can better determine where the positive of your population is and where the negative population is. Looking at the first part in A, we see PE versus FITC, and that's going to be the same across all A, B, and C. But it has no staining. There's no antibodies in the antibody batch, so it's just completely negative. So we just don't see any positive gates. That's why it's all like in the bottom right-hand side of the gate there. It's just all populated there. So this is just completely unstained everything. B is all of our antibodies except for the antibody of interest. And in this case, it's PE. So where the FMO bounds is in B, you see that line, that's where it's indicated everything that's negative for PE. So we just don't see any CD4 in that population. So this is what our absolute negative looks like. And then CD3 is our fully stained panel. It has CD4, CD3, CD8, all of those. So now we can determine, okay, this is where our positive population is. We can see at the top of C there. And then we have our complete negative gate, which is our FMO bound. And so everything below that line is considered negative for that CD marker. In this first article, it looks like they mostly looked at dendritic cells and breast cancer and some subsets of the dendritic cells. This is also an example of a gating strategy. So essentially they're taking, like their very first gate is your side scatter versus forward scatter. And if you remember at the beginning of the presentation, we talked about forward scatter is really looking at the size of the cells. Side scatter is talking about the granularity. So per literature, this is probably where our cells of interest are going to be. So this is where they gated out their cells, and then from this gate, they created this gate. And this is your side scatter wide versus side scatter area. And this is most likely their single cells. So with flows, you really want to focus on your single cells. You don't really want to look at doublets, which are most likely all of the extra things that aren't in our gate. And then from here, we took our side scatter A, and then DAPI is a viability dye. So it looks like they took the negative DAPI cells here, and then that's what they went with their negative. So then we have our CD45. Again, this is in humans, so NHP, they're cross-reactive, so CD45 is going to be the same. These are our leukocytes. So we're going to gate out our positive leukocytes here, and then we're going to use that here. CD14 is a monocyte marker, so anything up here is going to be positive for your monocytes. Anything over here is going to be positive for, it looks like they did CD3, CD19, CD56. And so they took the negative of these markers, but the positive, the CD14, and so they got this gate. And then from there, they looked over here. CD11C is a dendritic cell subset. So that's this, and they did it against the HLR-DR. None of their graphs have like their fluorochrome, so I'm not sure which fluorochromes they're using in these. But we can see our positive population for the HLA-DR and our negative, as well as our positive CD11C, and then our double positives over here. It looks like they took the double negatives and then the positive HLA-DR, and then they used the HLA-DR against CD123, which is another dendritic cell subset. And then they grabbed the double positives, so that's what they were gating out here. And then from here, they took the double positives, and then they did CD14, which is another monocyte marker. I'm assuming they were trying to gate out the rest of the monocytes, and then these were their dendritic cells. Monocytes are pretty common to use with dendritic cells just because there are monocytes that are derived from dendritic cells. So that looks like the type of gating strategy that they were going for here. So yeah. So like these are double positives, these are just positive for CD1C, and then our double negatives. And these percentages are just the percent of total, usually. So these are just the percent of the total are these 5. The 5.88% is percentage of your double positives of your total sample. And that's just kind of how you read those. Here's another article, another gating strategy. So they use forward scatter height and forward scatter area. They started with isolating out their single cells. So these are your singlets, and then these are all your doublets. And then from there, they did side scatter versus your CD45 V500. And then they gated out their lymphocytes and their monocytes. So 2 is monocytes, and then these are most commonly probably your lymphocytes, and these are probably granulocytes here. This is probably just some debris. CD14, so again we have our CD14 positive, and then our HLA-DR fit C. This is another dendritic cell study. They were looking at dendritic cells again, so that's what we're going to see here again. But so they're going to grab all of their dendritic cell subsets and their monocytes, and that's what they grabbed with this whole gate here. And these are just their double negatives, so they weren't interested in those. So they just grabbed these, and then they moved over here. So again, we have our CD14 APCH7. So that's a monocyte gate. And the CD16 is another monocyte marker. So they just grabbed all of the positive monocytes, and then they moved them over here, where they did against a dendritic cell marker. And then they were able to gate out, so like this is all of their double positive and positive for both markers. And it looks like they just pretty much continued just grabbing the positive populations just to get further subsets. So these are all different subsets in which they identified using this. Going back here, they used their double negative monocyte gates probably to identify their dendritic cells, which we can kind of see CD123 and CD91. Our double positive, or just our positives, CD91 they used here, and then they did gate out their CD123. That was just probably another value that they obtained. Yeah. And our last one, this one's a little bit simpler. Again, dendritic cells, it looks like they're single cells. They labeled theirs a little bit more to make it a little bit more easier to read. Forward scatter height, forward scatter area, got their single cells. And then from there, they used the viability dye. So then they gated out all of the live cells. And then from there, they gated out their CD45, so their immune cells, their leukocytes. And then from here, they did against the LY6GPE, and then they're double against the CD11B. And so they got a double positive for neutrophils. And so this one was a lot simpler than the other ones, but it still conveys just as much information. So they're able to get their percentages of their neutrophils and see what percentage of neutrophils comes from the whole sample. And here are my references. I hope you guys enjoyed the presentation. I know there's a lot of information, and I probably could have talked for a lot longer, but yeah, I hope you guys enjoyed it. Thank you.